Good evening and welcome to the European project session. First of all, I would like to thank the organizers for putting together such an extraordinary meeting. I would like to let you know that this session is organized in 5 talks. The first 4 by Roderick Guigo, Giorgio Bertorelli, and Cristina Roquet will cover different projects for sequencing biodiversity in different areas of Europe. The last talk by Claudio Cioffi will present a pan-European proposal that has been mentioned by Eric Jarvis. And this proposal aims at providing a comprehensive description of genomes of European species as a measure for preserving them. In terms of session organization, talks will be played back-to-back, followed by questions at the end. In this way, we hope to have some room for general discussion. Thus, in order to address the questions to the proper speaker, I could ask you to please write his or her name or some identification at the beginning of your question. With no further information, I think we should start with the meeting.
Okay, thanks for inviting me to present the Catalan contribution to the Earth Biogenome Project, the project that aims to sequence all eukaryotic species living in the Catalan territories. So this project started in October 2018, prompted by the EVP paper in the PNAS. We wrote a position paper by the Catalan Society of Biology, which is one society affiliated to the Institut d'Estudis Catalans, the IEC, which is sort of the National Academy of Science of Catalonia. Initially, we were proposing sequencing of all endemic species living in the territories, but we moved this later to a more ambitious project of sequencing all eukaryotic species. The paper was later endorsed by the whole Catalan Institution for Natural History, another society affiliated to the Institut d'Estudis Catalans. So in April 2019, we organized a local meeting with stakeholders, so research institutes, infrastructures, environmental and funding agencies to present and discuss the project and to obtain some feedback. And after the meeting and until the fall, we developed a number of actions. We set up a steering committee and were able to get some administrative support. So this is the members of the committee which we are in charge of actually starting the project. There is administrative support and there's also support for communication. We also started to set up the web page, which you can see here. And the most important thing is that we managed to seek funding. The seek funding is through a donation of Leandre Cervera Legacy to the Catalan Society for Biology. So Leandre Cervera was the president from the Catalan Society from 1935 to 1963. These were very difficult times. The society was illegalized in 1939 after the Franco victory in the Spanish War. And all the activities had to remain clandestine for almost 30 years or 25 years until they could be again open, publicly open in 1962 when Leandre Cervera resigned as a director. So he donated part of his personal legacy to the Catalan Society for Biology, and we are actually using these funds to start the project. In September 2019, we organized a symposium of biodiversity genomics. By the way, this is the name that we gave it, mostly to make the local community aware of the efforts worldwide. Many of the speakers that are present in the meeting today were also present in this meeting, and the name is leading the Earth Biogenome Project, the Tree of Life, etc., and many other projects. This is a picture actually of some of the participants taken at the IEC site, where actually the meeting took place. In January 2020, the IEC became an institutional member of the EVP. Here you can see it's among the members of the EVP, and now we are working towards becoming an affiliated project. In the first months of 2020, we put together a brochure to be addressed to the general public. So we actually want this problem and want to engage citizens very much in this project, and also a dossier to approach funding agencies. So this is part of the brochure that we developed, and this is the dossier. We also were able to find some small additional funding from the number of institutions, mostly the Barcelona Zoological Park and the Department of Agriculture from the government of Catalonia. This is some of the species in which the zoo was particularly interested. And everything, we had a good momentum, and everything was ready to set up a two-day meeting to really be the foundation of this project. The meeting was set for March 23-24. We had actually funding by a private local institution. So we actually managed to find a nice location, a medieval monastery in Catalonia. But as you may guess, at the end, a few weeks before, the COVID-19 crisis struck, and we had to cancel the meeting. This, of course, everything associated with COVID-19, as many other activities, was affected, and the project was really slowed down very much. We lost a lot of momentum. We tried to rebuild the momentum, and we substituted the meeting with 2 virtual meetings in June and July. I think in this particular, virtual meetings are very good in some cases. I think in this particular case, everything would have fed very much from personal interactions. Anyway, so during the meeting, we started the organization of the project and setting the working groups. So there are now 3 working groups: sampling and storing, sequencing and bioinformatics, and funding and outreach. These are the members that have volunteered to participate so far in these working groups. But of course, there are many people who are still to participate, and we are working on the organization and the governance of the project. So essentially, we think that the model in which the model is led by the Institut d'Estudis Catalans. So it's a scientific academy rather than a research institute. I think it's a feature of our projects, because this scientific academies actually capture the diversity of research and interests that is going on in the country. And of course, some of the groups have more experience in species cataloging and sample acquisitions, and some of the other groups are actually focused on the technology, on the data production, the sequencing, the bioinformatics. And one area that we have identified that we are not very strong is in the biobanking, and this is where we need to put really efforts into this project. We also announced a call to fund pilot projects. This was the announcement that I think was out just before the summer. I think that we were sort of surprised by the success of this call. We got 25 applications, which are currently being reviewed by some members of the EPP, which we thank them a lot. And the funding decision we announced in a couple of weeks. So we also discussed the name and the logo of the project, but we haven't reached yet a final conclusion of this. It's quite controversial. So why a Catalan biogenome project? Well, there is a clear reason. The project is led and funded by the Institut d'Estudis Catalans, the mandate of which is to promote science and humanities in the territories in which the Catalan language is spoken. This includes territories in 3 countries: Andorra, Spain, and France, and including actually the Balearic Islands. But of course, this is not the only reason. The Catalan countries are actually a biodiversity hotspot. This is just only in Catalonia, and there are many different habitats and types, which actually correspond to many of the main biogeographical regions that can be found in Europe, from Mediterranean, the land, then the water, the transition between the land and the water, the continental, the Euro-Siberian, and the Boreal Alpine. So in total, I mean, it is not, of course, this is just an approximation, but there are about 35,000, maybe 40,000 aquatic species living this is just in Catalonia, but probably it's a larger number if we include all the Catalan territories, which actually comprise about 1/4 of all European species that are known. Catalonia has some sort of degree of protection. About one third of the area has some sort of degree of protection, which also helps to maintain biodiversity. Second, the third reason is because there is a rich naturalist tradition in the country. The ICHN, the Institute for Natural History, was founded actually in the 19th century, and there are dozens of amateur, but some of which quite professional naturalist associations in which the project is also counting on. We have leading infrastructures that are needed for a project like that in terms of sequencing and computation. In Barcelona, there is the National Center for Genomic Analysis, which is one of the largest sequencing centers in Europe after the Wellcome Trust Sanger Center, and the Barcelona Supercomputing Center, which is one of the largest computational infrastructures in Europe. So what is the current status of the project? So we have a few poster projects underway. We are going to, as I said, announce the funding decisions on the pilot call very soon. We're working on the organization and to streamline the production pipeline. That's the most important thing. We are searching for additional, really core funding to make this project really viable. And of course, we are also looking at the future, right? Connecting with other projects. So this is some of the projects that are currently underway. There are 3 species that Tomás Marquès is actually involved in generating high quality genome sequencing using a variety of technologies, bio, nanopore, etc., etc. And of course, looking towards the future, we envision the Catalan project to be a node of larger projects, larger networks. We are particularly interested in contributing to projects in the Iberian Peninsula and the Mediterranean Basin, which are our natural biogeographical regions. We also, and let me finish with this, we think that we could contribute to the Earth Biogenome Project, not only by providing genome sequences of a species, but actually I think that we have a quite rich bioinformatics ecosystem in the country, and I think that we can develop methods that could be actually widely used across the project. For instance, I'm just listing a few examples. Cedric Notre Dame at the CRG is working in very, very large multiple sequence alignments of millions of sequences. Tyler Ragnoto at the CEGLAC is working with methods for genome assembly and annotation. Antonio Gabaldon at the Barcelona Supercomputing Center working on phylogenomics methods. All these methods could actually be of great interest to many participants in the Earth Biogenome Project. And with this, I'd like to finish, and I'd like to thank you and thank everybody that believe and contribute to the project. Thanks a lot.
Hello to everyone. I'm Giorgio Bertorelle, and I coordinate ENDEMIXIT. This is a project started this year where we plan to use whole genome polymorphisms to study and protect 5 endangered, iconic, and endemic species. 4 of them are considered good species and 1 is a subspecies. So let's see. The Marsican bear first. It's a subspecies of the brown bear, present only in a very small population in central Italy. It's 50 individuals left and isolated since several thousand years with a genome size of 2.8 gigabases. The Adriatic sturgeon, almost extinct in the wild, with a genome size of 2.0 gigabases, but this is a tetraploid species. The Ponza grayling, it's a butterfly with a small genome, present only in a couple of small islands in the Tyrrhenian Sea, the Ponza Islands. The Aeolian lizard, you can find this lizard only in a few volcanic rocks and islands northern of Italy, the Aeolian archipelago. The genome size is 2.2 gigabases. And finally, the Apennine yellow-bellied toad, distributed along the Apennine chain in Italy with the northern populations with very, very small sizes and quite big genome size, approximately 10 gigabases. We are a network of 4 universities. It's me in Ferrara, and then Claudio Cioffi is coordinator in Florence, Leonardo Congiu is coordinating in University of Padova, and Marco Gerdel is coordinating in the University of Trieste. Then we have several collaborators, 4 postdocs and 2 PhD students. So which are the main goals? Yeah, first of all, we would like to produce genomic resources for this species, producing de novo reference genomes with a mixed strategies and including also chromosome sequencing for the toad, where we have a big genome size. Then we will do genome resequencing in approximately 20 to 30 individuals per species. And the final total effort in terms of sequencing will be approximately 7.5 terabytes. But the core goal of the project is to use polymorphisms to understand the accumulation of deleterious mutations, the genomic load, in small populations and in control groups. And for us, control groups will be represented by either large populations of the same species or endangered, non-endangered sister species with very, very large population sizes. We will also estimate demographic histories and use genomic simulations to predict the effects in terms of load reduction and ancestry loss of genetic rescue strategies. We will also perform a couple of functional assays. In bare cell cultures, we'll try to understand the real effects of some mutations that were predicted to be deleterious and are fixed in this group. And in sturgeon crosses, we will study again the effects directly on the fitness of the offspring of some specific mutations we will predict to be deleterious. Finally, we will also disseminate our work step by step, the methods and also the results we obtain to promote the use of genomics in conservation biology, especially in Italy. Most of the samples are already available from previous studies, but we performed also some sampling last year and this year. For example, here in the first row, you can see the Aeolian lizard sampling in these volcanic rocks. And in the second row, there are some examples of the sampling of the toad, the bear, and the butterfly. As I told you, we started very recently, but we have already some results. For example, the assembly of the Marsican bear genome is quite good. We have good statistics for the PacBio sequencing of the in the Ponza butterfly. We succeeded in the first sturgeon reproductions with the hatchling of the eggs. And in the panel in the right bottom side, you can see some preliminary result of the functional assay in the bear cell cultures. Essentially, cells were transfected with ND5 gene with mutation typical of the Marsican bear, predicted to be deleterious. And this is a gene very important for the energy production in the mitochondria. And as you can see, the two curves correspond to Marsican bear and non-Marsican bears. So the performances here in terms of calcium uptake are different. And so probably these mutations are really affecting performances in terms of energy production. Finally, for our dissemination task, we have now a website. We have a blog in the website where we try to update as frequently as we can about our work, and we have also a Twitter account. So I invite you to have a look. Thank you very much for your attention.
My name is Camila Mazzoni, and I'm a group leader at the Leibniz Institute for Zoo and Wildlife Research. And I also have a role as the bioinformatics head at the Berlin Center for Genomics in Biodiversity Research. I would like to first of all thank the organizers for the invitation to give a talk. And in this presentation, I will briefly explain the experience of my group with the Vertebrate Genomes Project. But I would like to take this opportunity to discuss about an emerging German initiative for biodiversity genomics. In these maps, we can have an idea of how extensive and also decentralized biology-related research is in Germany, including tens of institutes and graduate schools dedicated to ecology and evolution. Germany also hosts an invaluable natural history collection with more than 70 million specimens present in 6 of the almost 300 museums. In January of 2019, the German Science Foundation, DFG, has founded the NGS Competence Network, including 4 competence centers located in different parts of Germany, holding advanced genomic technology. Despite all this available infrastructure, we haven't been able to identify any large-scale biodiversity genome project based in Germany. In contrast, there is extensive participation of German research groups in several international large-scale genomics initiatives. I would like to call attention to the Vertebrate Genomes Project, which aims to sequence high-quality genomes for all vertebrates. Since the end of 2017, my group in Berlin has joined Jean Myers and his team at the Max Planck of Molecular Cell Biology and Genetics in Dresden to sequence genomes from 3 different tropical vertebrate species as part of the VGP. The genomes were almost entirely produced within Germany, from DNA extraction to sequence assembly. However, as part of the VGP consortium, every assembly goes through curation by the Sanger Institute and is part of numerous discussions within the VGP assembly group. The assembly group, similarly to other topic-oriented groups within VGP, puts together leading worldwide experts in the area. The expertise accumulated within the consortium helps establishing and maintaining the consistency in genome quality across species and prevent researchers from struggling with the same problems over and over again. CBG from Dresden is one of the 3 sequencing hubs of VGP, playing a major role in the project. The CBG and the Center for Systems Biology Dresden participate in 3 major vertebrate genomic sequencing consortia. The 2 institutes are part of the Dresden Concept Network, one of the 4 NGS competence centers funded by the DFG, which is currently finalizing more than 70 high-quality vertebrate genomes distributed across the different projects. When we joined the VGP initiative as a single research group, we asked ourselves in which way we could support VGP other than sequencing our own genomes of interest. First, we took advantage of the bank of tissues of the pathology section from IZW, obtained from necropsies from zoo and wild specimens. The high-quality tissues are suitable for ultra-high molecular DNA extraction. In parallel, BGenDiv, a small-scale genome center dedicated to biodiversity, is located at the Freie Universität Berlin, one of the largest universities in Germany, which hosts a bachelor and master degrees in bioinformatics. With a group of trained bioinformatics master students, we gave the first steps towards building an army of volunteers that will help assembling thousands of genomes across the years. Our students helped substantially in the beginning, testing and automating the assembly pipeline in the cloud-based platform called DNA Nexus. At the start of this year, a branch of VGP was formed as an European initiative to create an European reference genome atlas. A large movement is currently in place to convince the European Parliament and European Commission about the importance of sequencing genomes from all life in Europe. In parallel to the ERGA initiative, a number of Germany-based researchers started a group to join researchers and institutions interested in supporting ERGA. The next step of the ERGA.Germany is to create a strategic plan to connect research institutes, museums, sequencing centers, and bioinformatics teams to create a national-based network that can be the basis for this and other large-scale genome projects connected to biodiversity. If you are a Germany-based researcher, please join ERGA.Germany discussion group, and if you have any questions, please write to any of the starting members of the initiative. Thank you very much for your attention.
Good evening. I will present you today the PhyloAlps project, which originated in the Laboratoire d'Écologie Alpine 10 years ago. The target organism of PhyloAlps are all the species and subspecies of vascular plants occurring in the Alpine arc at all altitudinal levels from foothills to nidal zones. As a reference list, we adopted the checklist of Flora Alpina, an integrated work by Eschimann and colleagues. According to this work, in the Alpine arc there are nearly 4,000 plant species. This is a huge number of taxa, and thus the sampling required high-quality expert knowledge to collect them on the field. Thus, to tackle the first crux of the project, We built a collaborative network of botanists working in French organizations called Conservatoire Botanique and in national alpine parks, plus Swiss researchers and botanists. We then established the protocol sampling. First, collect leaves of a single individual and introduce them in silica gel tubes to quickly dry them. Then collect an entire individual to make an herbarium voucher in order to be able to check potential errors of identification and recall locality information and update the online list of non-sampled taxa. Concerning the sample strategy, we did several sampling campaigns in hyper-diverse locations of the Alps with different ecological settings and evolutionary and biogeographic background. The following years, we followed a divide and conquer strategy in which each botanist sampled lacking species in nearby areas. We soon realized that there were controversial taxa for which we needed to reach a consensus on species delimitation. To solve this issue, we did a meeting with all the botanists. In the majority of cases, we agreed to group uncertain taxa in species aggregates. Concerning the sequencing, we considered that the most cost-effective strategy was shallow shotgun sequencing, which means to randomly sequence a certain percentage of total genomic DNA. Given that the chloroplast and mitochondrial genomes are present in a much higher number of copies in a cell than the nuclear one, the full sequence of these genomes can be retrieved with low-cost shallow sequencing. The full sequence of the chloroplast genome is very useful in many fields of biodiversity research. For instance, it allows to conduct phylogenomic analysis at varying taxonomic levels from closely related species to the whole plant kingdom, resulting in highly resolved phylogenetic trees useful for evolutionary studies. In addition, this genome sequence is an excellent source of information for DNA and metabacoding studies. Current standard DNA plant barcodes are often difficult to recover from environmental DNA, which is highly degraded. Extending the concept of DNA barcode to the whole chloroplast genome would greatly increase the power of to discriminate species plants in metabacoding studies. Given the high volume of PhyloAlps data, we needed to build an automated workflow to treat the sequence raw data. To do so, we developed a program that makes automatic de novo assemblies of organelle genomes, resulting in a single circular conjugate in less than half an hour, using a single core for 70% of our taxa. In addition, we developed Organote, which automatically annotates chloroplast genomes based on a created database. Our current version is able to annotate 90% of the genes commonly found in chloroplast genomes. So, which is the current state of the project? Nowadays, the sampling is finished, and we are organizing a reference herbarium with all collected vouchers. Sequencing has been performed for the majority of samples in Genoscope, and we have developed a web database that in the near future will allow public access to molecular data, collection, and ecological information of the taxa. Given the advanced state of the project, we hope that next year we will be able to publish a highly resolved phylogenomic tree linking all the plants of the alpine plant biome, which will be used to study diversification dynamics in sky island systems, as are mountains. The deluge of data generated by PhyloAlps has opened the door to many research lines currently being developed. For instance, Jan Smitka has conducted a PhD thesis on the diversification of multiple European plant clades. I am working on plasmodium evolution in parasitic plants. Eric Coissag and Franz Deneux are investigating different aspects of genome evolution. And Wilfried Thuillier, Luc Géry, Carcaillet, and Nick Zimmerman are working on the use of PhyloAlps database for metabacoding studies and conservation planning in the Alps. Thanks for your attention.
Hello, everyone. My name is Claudio Cioffi, and I'm going to present a pan-European initiative on the importance of genomics and reference genomes in particular to biodiversity conservation and ecosystem services. Biodiversity is central to maintaining ecosystem functions, and therefore it is widely recognized as a fundamental asset for many sectors of the global economy. Unfortunately, biodiversity is under threat worldwide. Based on the 2019 UN report on natural decline, 1 million out of 8 million species on the planet are at risk of extinction, and many could go extinct within decades. This includes 22.7% of European species, of which 17% of mammals, 12% of birds, 20% of reptiles, and 24% of amphibians. There is evidence of decline for freshwater fishes, marine fishes, invertebrates, and approximately 35% of plants. Now, in her speech at the European Parliament plenary session in November 2019, as President-elect of the European Commission, Ursula von der Leyen introduced the agenda of the new Geopolitical Commission aimed at reinforcing the European Union's role as an international leader in the transition to a healthy planet. Indeed, the preservation of biodiversity and restoration of European ecosystems are included in the 6 political guidelines and priorities that will shape the upcoming working program of the European Commission. Conservation of ecosystems and biodiversity is, in fact, a crucial priority for the essential services they provide to agriculture, fisheries, aquaculture, food security, water and air filtration, natural disaster risk, pests mitigation, climate regulation, and also discovery of new medicine. Now, biodiversity implies genetic diversity. The EU biodiversity strategy highlights, for instance, the preservation of traditional variety of crops and breeds. Domestic variety of plants and animals tend to show a high degree of genotypic and phenotypic adaptation to local conditions, with the resulting decline in genetic variation. To this respect, wild relatives of domesticated breeds are particularly important as they present critical reservoirs of genetic and trait diversity that may provide resilience against future climate change, pests, and pathogens. Genomics is key to preserving biodiversity. For instance, in biodiversity restoration, the function of ecosystems that have been restored needs to be assessed and monitored to confirm success of restoration efforts. However, many biotic components of ecosystems are either difficult or impossible to survey using field-based monitoring techniques. And for instance, because rare species can be elusive or difficult to distinguish. And here, genomics can very precisely characterize all the diverse taxonomic groups and monitor trends and in their distribution and abundance. Genomics can also help understand past and present demographic trends of populations and predict if, how, and to which extent species important to ecosystem services will cope with habitat changes, how their distribution can respond to the ongoing shifts in habitat quality, and what their level of resilience will be in terms of, for example, susceptibility to pathogens and adaptation to a changing environment. Understanding features of the genome related to the ability of organisms to respond to such challenges will help better understand how to preserve ecological community and ecosystem functions. Now, genomics typically samples thousands of molecular markers distributed across the genome of a species, providing a high resolution of genetic and functional diversity. Historically, the genetic study of any living species starts from the generation of a reference genome, which is a high-quality sequence for a chosen individual of that species. While what constitutes a reference genome has evolved since the first genomes were sequenced in the late 1970s, it has remained consistent that such references informed any subsequent genetic study, both in the planning phase and in the analysis and interpretation of results. Now, in August 2020, so a few months ago, a position paper signed by representatives of 39 European institutions belonging to 16 EU countries, as well as 9 institutions in 6 associated countries and 2 extra EU partners institutions in the USA and UK. This position paper was presented to the European Commission's Directorate General for Research and Innovation. The position paper argues that the upcoming Horizon Europe research budget should include a large genomic initiative, tentatively named the European Reference Genome Atlas, or ERGA, to generate reference genomes representing the European biodiversity, including species of importance for agriculture, fisheries, ecosystem function and stabilities, pests, and of course, endangered species. The idea of an ERGA came up last year in November 2019 during the European PacBio user group meeting held in Milan, in Italy, and was soon after discussed in January 2020 at the Vertebrate Genomes Project meeting held at the Plant and Animal Genome Conference in San Diego in California. The idea of an ERGA builds on growing genomic consortium efforts in European Union and non-EU countries, first of all the Vertebrate Genomes Project, or VGP, and others like the Earth Biogenome Project, the Darwin Tree of Life Initiative, the Bird Genome 10K, the BAT1K, and other more recent EU projects such as the Catalan Biogenome Project and the Italian Population Genomic Project for Endemic Species. Now, a common mission of this project is to generate high-quality, near-complete, and error-free genome assemblies of all species within a group, like vertebrates for the VGP, or a geographic location like the UK for the Sanger-Darwin Tree of Life, to address fundamental questions in biology, disease, and conservation. In our view, the ERGA initiative should integrate with and complement these initiatives as far as European species are concerned, potentially adopting laboratory and bioinformatic pipelines developed, for instance, by the Vertebrate Genome Project. Now, as a whole, the EU currently lacks a coordinated, systematic financial and institutional leverage required for supranational initiatives of this kind. Yet we believe that with the appropriate support, cross-country infrastructure and coordination, the fundamental goals of the ERGA are within reach and will complement ongoing individual efforts by countries and projects. Similar to the influence that the Human Genome Project had in the last 2 decades, we argue that the Atlas will set solid foundations for hundreds of specific projects for conservation assessments, biodiversity monitoring, and restoration efforts to come. Having a long-lasting impact within the upcoming Horizon Europe program, this initiative will also help establishing the EU as the world leading union in the area of genome research applied to biodiversity conservation and restoration. The full position paper is available at vertebrategenomeproject.org/ERGA, as you can see in this last slide. And we encourage other institutions interested in applying the study of reference genomes to EU biodiversity and ecosystem conservation to support and join our initiative. And we also invite the audience to join our signatory list and our mailing list in case you wish to be kept in the loop. Thank you very much.
Okay, thank you very much to all the speakers. I'll see if we have a few questions. The first one is for Roderick, and the question is, are you able to copy and paste guidelines, protocols already developed by, for example, the EVP or the Darwin Tree of Life, or you find you need to make your own?
Well, I think that's actually the word, copy and paste, copy and copy. And we are totally aligned, or we plan to be totally aligned with the protocols from the EVP regarding collection, storage of samples, sequencing, informatics, and data release. Actually, we've been in touch. I haven't mentioned this, but I think that I should really thank an immense thanks to Mark Blackster from the Tree of Life, who has been advising us a lot on how to really organize the project, and also Harris Lewin, who are actually helping us a lot. So essentially, the plan is to align as much as possible. We don't need to reinvent anything that is working. Of course, there are issues that may be peculiarities of our project. One thing is regarding prioritization of species. Of course, we would like to prioritize species from taxa that have not been well captured so far, but we are not too much worried about duplication of species. I think that species, the same species, is different in different places, and I think that having different genomes from species living in different areas may be actually also interesting. Second, of course, there is, for instance, in Catalonia, there is an effort now to revive cultivars that have been abandoned. So that's something that maybe an emphasis on agricultural genomics may also be a peculiarity of our project. And third, as I said, I think that we had expertise in different areas of informatics that could contribute to the protocols and to the pipelines of the EVP, and actually would like to contribute to them. Not only informatics, actually our group has developed a method now, a CapChart method, to sequence full-length RNA sequence, which actually after the Harris talk, I think it could be of particular relevance. So we think that we would like also to contribute in the other direction to the EVP, but essentially it is copy and paste. I don't think there is a need for us to do anything different unless it's clearly superior.
There is another question that I guess is for Christina, I'm not sure, it says, are the stored biological samples up for genome sequencing? Christina.
Hi. Well, they are. We collected them in silica gel tubes. We should check if they are really suitable for high molecular weight sequencing. In case it's not, at least we have relevant information on where these plants can be found. I would like to remember that in the European Alps, there are over 500 plants that are endemic to the Alpine regions. So at least this is for sure an information that we had.
Okay, then I have a couple of questions for Claudio. Can individuals or labs be involved in prioritizing species selections? Are we not quite at that stage yet? So how do you see this? How do you see this effort, European effort?
Yeah, I think of course both individuals and labs can be involved in prioritized species selection, but the other answer is that we are not quite at that stage. So far, we are basically collecting both suggestions and I'd say subscription applications from different groups that would like to be involved. But we have first to see whether at the European level our initiative is received or not. So before going into details on who will do what and how many say institutions will be involved and what will be the prioritization of species selection, we will have to probably make sure that our initiative is likely, say, to be funded by the European Union. But of course, everybody will have a saying in our initiative.
Then there's another question to Claudio as well. How we promote genome projects in conservation policy, considering that genetic diversity is still overlooked in national conservation programs?
This is a good question. Perhaps Giorgio, it would be more kind of able to answer that. Yeah, say something.
I think that is, of course, very difficult to promote the use in applied conservation of genetic and even more genomic data. But the project I described, and Claudio is involved as well, is just an attempt of that, doing 5 species that everybody know about in a country and using genomic data to suggest strategies and showing how to do it step by step. At the end of our project in 2023, I hope that there will be a little bit more awareness about the use of conservation genomics, of genomic data and conservation. So this is just a way to proceed, doing projects and showing how this data can be useful.
I think there's another question that can be for Claudio or for any other of the speakers. This says, how do you realize a specimen long-term storage?
How do we realize what?
How can we keep the samples for long-term storage? How can we store the samples for a long term?
I think most of the labs have ultra freezers -80 and no, I mean, very long-term storage are generally done by institutions like in California, they have the project like the Frozen Zoo, in which they keep samples, let's say, at liquid nitrogen temperature. If I understand the question, is that?
I guess that's the question. Yes. And then there's another comment says, when working in geographically restricted areas, how do you deal with reference sample choice if there is potential interspecific hybridization? Maybe that's also a question for like a global question for any of you. How to deal with interspecific hybridization?
Interspecific, you mean? Yeah, like sister species that hybridize, you mean, I guess, or? I mean, we are facing exactly problems like this one, because close species, especially when they are sort of invading environments, also because of human translocations, hybridize with local species if they are close enough. So the idea, if you want to protect a local species, an endemic species, is to use sort of pure samples, you can find them, to produce the reference genome. And then, of course, the genomes can be very, very useful also to understand better the hybridization process and understanding also if there is a sort of species reversal, so that two species come together and they hybridize until the point that you don't have any more the possibility to detect them. So all this process can be much better understood when you have genomic data. Not just the reference genome, of course. The reference genome is just the point where you start, but then you have to study resequencing data, whole genome sequences from larger samples of individuals.
And if I can add to that, I think in that case, so when you're choosing a reference genome or a sample for building a reference genome, a preliminary study would be necessary, because it does happen that you assume that a specimen is pure, and then when you sequence the genome, it's actually not. So
right.
Preliminary analysis, maybe with reduced representation or any sort of analysis that you can do in a short term and not with large costs, could help a lot in selecting the most appropriate sample for a reference genome.
Yes, you're right.
Also, if I can comment on this, I mean, the example of the human genome may be valid to some extent. So in the human genome, we're transitioning from a linear representation of the genome to a graph representation of the genome to be able to capture with a single representation all the species, in this case, intraspecific variability within the species. So I actually don't know what are the plans for the EVP or Tree of Life, but I mean, this graph genome has some utility in the case of the human genome to represent in a single structure the variability of a given species.
I don't see more questions. I don't know. Oh, well, hold on. There are some comments now about that. Natural hybridization is probably much more common than zoologists had thought. It's not a problem, but it's part of the EVO process. And then also there is another question now. Is it possible to include assays for intraspecific genome variations for the species included in the project? Can you? That's a question for Claudia, I guess.
The last one, you mean?
Is it possible to include some specific assays of intraspecific genome variation for the species included in the project.
Well, yes, one of the projects that we show, probably the one that Giorgio talked about, that deals with that. It's population genomics and intraspecific genome variation. If that can be included in the project, that could be quite useful, but that depends on the on the amount of fund that one has at his her disposal. If the project is well funded, then one can move from reference genome also to intraspecific genome variation. So we will have to make some, if the project is well received from the EU, we'll have to make some decisions on that. But if we have enough funds, the intraspecific genome variation could be considered as well.
Well, I can talk about my own experience with the VGP, because it does happen that many people connected to the project have their own projects aside, also in conservation genomics. And they do the reference genome with the help from the consortium, because this is apart from being a long and complex process, it's also quite expensive still. But then the groups that are interested in the conservation genomics side, they then participate resequencing like samples that are representative or that are relevant for the question. So there are many different ways of doing this, and really it depends on the priority of the project. If it's to cover the largest range possible, phylogenetically speaking, or if it's for facing the most urgent cases of threatened species. So I think that's something that should be decided. What is the priority, and then what to do next?
Yeah.
Okay, I don't know if another question now for Camila. Could you elaborate on how the German project will work?
I mean, I wish, yeah, I think we are at the beginning of this process as well. As much as Claudio said, ERGE is in the beginning, it's the same. So Germany has an amazing infrastructure in place, but it's not exactly coordinated, and it's to a very little scale dedicated to biodiversity. So that's something that we can potentially change, but we have to act not only scientifically, but also politically. And I think by joining researchers from across Germany that are enthusiastic, I think that's what's going to help us make up this plan. And as already said by the others, we're going to take the lessons from the large projects that are already pretty much advanced. So, but I cannot say more than that.
Okay, so any of you want to comment, say something else? Because I think it's time to close. Thank you all for this, the speakers and participants, for this very interesting and inspiring session. As I can see from your answers, it looks like we are still at the beginning of many issues. So I guess we still have to talk a lot to try to achieve coordinations at different levels, and that will be an important thing to do from now on. I just want to remind you that all this talk will be posted at the Biodiversity Genomics website for a few days. I'm sure that this, as I said, this is just the beginning. Many more European and international projects will arise in the near future. We are really looking forward to that. But because it's clear to us, I think it's clear to all of us that genomics is essential to preserve biodiversity, we hope also the large genomic initiatives, such as the one presented here by Claudio, the European Reference Genome Atlas, will serve as a platform to obtain appropriate support, maybe cross-country infrastructures. And I think what we can the take-home message from this meeting is that, in my opinion, what we need is really work in a coordinated manner across Europe and probably across the international, globally. And that's a thing that I think we should do in the near future. So thank you very much to all, and we'll see you the rest of the meeting.
Thank you.
Thank you.
Thank you. Bye-bye.
Thank you.